Hi everyone, my name is Paolo Yannick de Matera. In this video presentation, I will be discussing the perpetual of life and interaction between plants and animals. My speech is all about earth and science, which will be informative and educational, so I hope you will like my presentation. What are the different ways plants can reproduce? Flowering plants reproduce sexually through a process called pollination. Flowers contain male sex organ called stamens and female sex organs called pistils. The anther is the part of the stamen that contains pollen. Pollen contains the male gametes. Pollen must be moved to a part of the pistil called the stigma for production to take place. What are the different ways animal can reproduce? Asexual reproduction. Asexual reproduction occurs in prokaryotic microorganism, bacteria, and in some eukaryotic single-celled and multi-celled organism. There are a number of ways that animals reproduce asexual. Fission. Fission, also called binary fission, occurs in prokaryotic microorganism. In some invertebrate multi-celled organism, after a period of growth, an organism splits into two separate organisms. Some unicellular eukaryotic organisms undergo binary fission by mitosis. In other organisms, part of the individual separates in form of second individual central disc. Some sea anemones and some coral polyps also reproduce through fission. Bonding. Bonding is a form of asexual reproduction that results from the outgrowth of a part of a cell or body region, leading to a separation from the original organism into two individuals. Bonding occurs commonly in some invertebrate animals such as corals and hydras. In hydras, a bud forms that develop into an adult and breaks away from the main body, as illustrated. With this, in coral bonding, the bud does not detach and multiplies as part of a new colony. Fragmentation. Fragmentation is the breaking of the body into two parts with subsequent regeneration. If the animal is capable of fragmentation and the part is big enough, a separate individual will grow. Parthenogenesis is a form of asexual reproduction where an egg develops into a complete individual without being fertilized. The resulting offspring can be either rip-lobed or dip-lobed depending on the process, and the species parthenogenesis occurs in vertebrates such as water fleas, rotifers, aphids, stick insects, some ants, wasps, and bees. Bees use parthenogenesis to produce haploid males, drones. If eggs are fertilized, diploid female develop, and if fertilized eggs are fed special diet, so-called royal jelly, a queen is produced. Sexual reproduction. Sexual reproduction is the combination of usual haploid reproductive cell from two individual to a form third asexual diploid offspring. Sexual reproduction produces offspring with novel combinations of genes. This can be an adaptive advantage in unstable or unpredictable environments. As humans, we are used to think of animals as having two separate sexes, male and female, determined at conception. However, in the animal kingdom, there are many variations of this theme. How do offspring inherit the traits of their parents? Chromosomes. Humans typically have 46 chromosomes in each cell of their body, made of 22 paired chromosomes and 2 sex chromosomes. These chromosomes contain between 20,000 and 25,000 genes. New genes are being identified all the time. The paired chromosomes are numbered from 1 to 22 according to the size. Chromosomes number 1 is the biggest. These non-sex chromosomes are also called autosomes. How we inherited characteristics. Parents pass on traits or characteristics such as eye color and blood type to their children through their genes. Some health conditions and disease can be passed on genetically too. Sometimes one characteristic has many different forms. For example, blood type can be A, B, or AB or O changes or variations in the genes of for the characteristic cause these different forms. Dominant and received genes. The most common interaction between alleles is a dominant recent relationship. An allele of a gene is said to be dominant when it effectively overrules the other recessive allele. How does DNA allow to transfer of genetic information? The flow of genetic information in bacteria, archaea, and eukaryotes. The primary roles of DNA is to store heritable information that encodes the instruction set required of creating the organism in question. While we have gotten much better at quickly reading the chemical composition, the sequence of nucleotides in genome, and some of the chemical modifications that are made to it. We still don't know how to reliably decode of the information within and all of the mechanism by which is read and ultimately expressed. Genotype to phenotype. An important concept in the following sections is the relationship between genetic information, the genotype, and the result of expressing it, the phenotype. These two terms and the mechanism that link the two will be discussed repeatedly over the next few weeks or starts becoming proficient with using the vocabulary. Genes. What is the gene? A gene is a segment of DNA in an organism genome that encodes a functional RNA such as RNA or tRNA. 8. A protein product, enzymes, tubulin, ink. A genetic gene contains elements encoding regulatory regions and a region encoding a transcripted unit. How does DNA allow the production of proteins in the body? How do genes direct the production of proteins? Most genes contain in the formation needed to make functional molecules and called proteins. A few genes produce regulatory molecules that help the cell assembly proteins. The journey from gene to protein is complex and tightly controlled within each cell. It consists of two major steps, transcription and translation together. Transcription and translation are known as gene expression. Can DNA be manipulated? If yes, how does it work? If not, why can it be done? DNA and RNA extraction. To study and manipulate nucleic acids, the DNA and RNA must first be isolated or extracted from the cells. This can be done through various techniques. Most nucleic acid extraction techniques involve steps to break open and the cell use enzymatic reaction to destroy all macromolecules that are not desired, such as degradation of unwanted molecules and separation from the DNA sample. Cells are broken using as lysis buffer, a solution that is mostly detergent. Lysis means to split. These enzymes break apart lipid molecules in the membranes of the cell and the nucleus macromolecules and are inactivated using enzymes such as proteases that break down proteins and ribonucleases are interactive to break down RNA. The DNA is precipitated using alcohol. Human genomic DNA is usually visible as a gelatinous white mass sample can be stored at 80% for a year. Do genetically modified organisms or GMOs exist? If yes, cite an example and describe how the organism is produced. If not, explain why they do. GMOs in agriculture. Genetically modified foods were first approved for human consumption in the human states in 1944, and by 2014 to 15, about 90% of the corn, cotton, and soybeans planted in the United States were GM. By the end of 2014, GM crops covered nearly 1.8 million square kilometers of the land, more than 2,000 countries worldwide. The majority of GM's crops were grown in the Americas. GMOs in the medicine and research. GMOs have emerged as one of the mainstay biomedical research since the 1980s. For example, GM animals models of human genetic disease enable to research to test novel therapies and to explore the role, candidate, risk factor, and modifiers of disease outcome. GM microbes, plants, and animals are revolutionized in production of complex pharmaceuticals by enabling the generation of safer and cheaper vaccine and therapeutics. Pharmaceutical products range from safer and recombinant habitats B vaccine produced by GM bakers used to injectable insulin for diabetics. Produced in GM integration biotrail and factor 8 for homophilics and tissue filaminogen activator. Both of each are produced in GM mammalian cells grown in laboratory culture. And that is for all, and thank you very much to hear my presentation. I hope you like it, and thank you very much.